Bismillahirrahmanirrahim. This is the second video on the topical paper 4 questions on the chapter on homeostasis. Question number 1. Some people have a condition called diabetes and type 1 diabetes. The pancreas does not produce enough insulin. Figure 7.1 shows the blood glucose concentration of a type 1 diabetic person and a non-diabetic person. So this is a diabetic person, which we have a graph like this, the one which I'm just drawing for you. And the non-diabetic person has a graph which is like this, and so I'm just giving it a color. So you have the blood glucose concentration on this side, which is in milligram 100 cm cube, and the time in hours is on this side. So describe the results. Now I always tell you describe is no biology. So you do not give me any biology, you just give me a description of what is happening in the graph. Is it increasing? Is it decreasing? So describe the results. So you have to give me both about the non-diabetic and the diabetic, and you sort of just compare the two graphs. When do they peak? How do they start? Is there a sharp increase or a gradual increase or a sharp decrease or a gradual decrease? These are the wordings that you need to use, and this is the type of English I expect you all to be writing in your A-level biology paper. So describe the results shown in figure 7.1. Now, if you look at the answer, describe is just something which every student should get the full marks in that, because there's no biology. If you don't even know this chapter, if you haven't even read this chapter, you can do this part of the question. So describe the results is first blood glucose concentration rises in both. Well, it makes sense, rises in both. So this rises as well with the drink. As soon as they've taken the drink, the glucose drink was taken at zero, both of them rise. So you have to look at the common factors. Then diabetic always has a higher blood glucose. They're all throughout this experiment, diabetic has a higher blood glucose, while the non-diabetic has a lower blood glucose. So something which is common in both and which you can do a comparison. Like if I say, oh, compare so and so with so and so, X person with Y, and you say both wear glasses, one is taller, one is shorter, or one is fatter, or one is thinner. So you do a comparison. So diabetic always has a higher blood glucose concentration. That's again something we didn't know any bio. Then the diabetic has a higher peak. So diabetic has a higher peak in the blood glucose concentration. And the second, the third, fourth mark scheme point was diabetic blood glucose concentration has a slower fall. You can see this point here, it's a slower fall, you see. While this person, it had a sharp fall. And then, of course, we come on to figures and units. Now, this is the important thing, what I want to talk about, the figures and the units. So you have to take 2 points at the x-axis. So I took 1 hour and I took 2 hour. And then in 1 hour, I've written down the points. So the person who was non-diabetic, it was 100 at 1 hour. And I'll just rub off all these lines so that then you all can see these points. So it was 100 at 1 hour in the non-diabetic, and then at 2 hours it was 108. So you see the figure here at 100, you have to see it here. I like you to just see a line here, and at 2 hours. So this is the non-diabetic person, right? And then I give you the figures for the diabetic person, and the diabetic person was of course Changad, and he or she, I don't know if it was a he or she, I'm just assuming certain things, and was 205 here, and then it was 192 here. So you just had to read off figures from the graph, but the important thing was that you had to give me the units. If you didn't give me the units, I'm not going to give you any marks. So you just wrote this, what I've written here, then I won't give you any marks. But if you wrote this units in with each of these, that this was the unit 100 milligram 100 centimeter cube and 205 milligram per 100 centimeter cube. So if you gave me the units, I would give you a mark for that. Otherwise, I mean, you're Changad, you won't get any marks. So figures and units. This is the describe question. Then coming to the part 2 of the question, it says name the location of the receptors in a non-diabetic person that detect change in the blood glucose concentration. Now, look at the different answers which are available to you. There are two reasons for that. Now, of course, I would make a question out of that. Beta cells is also allowed, pancreas is also allowed, islets of Langerhans is also allowed, and hypothalamus is also allowed. Now, why is the hypothalamus allowed? Because you see, if there is more glucose, what will happen to the osmotic potential? And that is detected by osmoreceptors in the hypothalamus. So that is why they have allowed you all these possible answers. More the glucose, if you take a glass of water and add 10 teaspoons of sugar in it, well, it's going to become very sweet. So it's going to less free water molecules. So we said the water potential of blood is sort of sensed by the osmoreceptors in the hypothalamus. So that's why all these answers were allowed. Beta cells was allowed, pancreas was allowed, islets of Langerhans was allowed, and hypothalamus was allowed. Then name the homeostatic mechanism by which blood glucose concentration is maintained. There's only, of course, one homeostatic mechanism, which everybody should know. Is the negative feedback is part of the syllabus point. If you've gone through when you've revised your paper 4, you must go through the syllabus point wise and see that you've understood each and every point of the syllabus. That is what they're going to ask you in the exam. Then coming to the next B part of the question, the urine of a non-diabetic person does not contain glucose. A person with type 1 diabetes will excrete glucose in the urine. A reading of the concentration of glucose in the urine can be estimated using a dipstick, glucose in the urine, enzyme A, gluconic acid plus hydrogen peroxide. Then hydrogen peroxide plus colorless chromogen, then enzyme B, water plus colored chromogen, and that is the color which you're going to compare with the color standard. The higher the concentration of glucose in the urine, the darker the color on the dipstick. So we have different colors with which we are going to match it. Name enzyme A and B. A is glucose oxidase, B is peroxidase because it's going to react with the hydrogen peroxide and is going to react with the colorless chromogen, which is going to change a color. An electric biosensor can be used to measure the glucose concentration. An electric biosensor can be used to measure the glucose concentration in a drop of blood. Suggest one advantage of using a biosensor and one advantage of using a dipstick to measure glucose concentrations. So, biosensors are very rapid, there's immediate reading in 2 seconds, more accurate, precise, numerical value, shows the current blood glucose, and the machine is, of course, reusable. The dipstick is non-invasive, it is painless, it is easy to use, less risk of infection, and it is cheap. So that is the points that you need to remember. So that's what I say. If you have done the topical of any chapter, you've covered nearly the entire syllabus on that chapter. And that is how I advise you to, when you study, first you do the revision and then you do at least 20 to 25 questions on that chapter. And you will see it, the same things come again and again. The same topics come again and again. So if you revised a chapter and you've done all its questions, well, I'm sure you're going to ace the exam. Part C, describe the role played by insulin in the control of blood glucose concentration. Number one, binds to receptors on liver/muscle cells, GLUT4. Now, 4 was not necessary, it's in brackets, so GLUT4 added or increases the muscle cells' permeability to glucose. Or you could have said GLUT4 added to membrane of muscle cell. Or you could have said GLUT4 increases the muscle cell permeability to glucose. That's all the same thing, means 1 mark scheme point. Then this will increase the rate of diffusion of glucose into liver cells. Then more respiration of glucose, so more ATP will be formed. Then causes glycogen synthesis, because more the glucose entering, it has to be converted to glycogen, which is an insoluble, compact molecule. Then more causes lipid synthesis, because naturally if you eat a lot of sweet stuff, you're going to get fat because lipids are going to be formed. Fat is deposited under the skin, and the blood glucose concentration will decrease. So 7 mark scheme points for a 5 mark question, and you have to give me any 5 to get the 5 out of 5. So unless you have been a careful student and you have remembered everything and you have memorized it, only then you're going to get the 5 out of 5. Not everybody who's just going to write 1 or 2 sentences and say, okay, well, I don't remember much of this. Question 2, figure 5.1 is a photomicrograph of a part of the cortex of the kidney. And you can see this whole thing is the glomerulus, and you can see the space it is, which is the Bowman's capsule space. So on figure 5.1, use label lines and letters to label the glomerulus and the lumen of the Bowman's capsule. Now, so this would be the lumen, this would be L, and the glomerulus would be anything from here. So that got you 2 marks because this is the space. This is what we must pick out on a photo micrograph, and that's a very important part of the mark schemes. Then it says during ultrafiltration, components of the blood in the glomeruli with the relative molecular mass greater than 68,000 are preventing from passing into the Bowman's capsule. Now name the structure that acts as this filtration. Now, everybody knows that basement membrane. So the basement membrane is the one which is going to prevent it. Everything else, so the endothelium of the capillaries, of course, they've got more pores in them as compared to normal capillaries. So everything come out except the basement membrane, which is going to allow it not to come out. B part of the question. The glomerular filtration rate GFR is the rate of flow of filtered fluid through the kidneys per unit time. The afferent arterioles supply blood to the glomerulus of each nephron within the kidney, and the efferent arterioles take blood away from each glomerulus. The lumen diameters of the afferent and efferent arterioles have a large effect on the GFR. Normally, the lumen diameters of the afferent and efferent arterioles are different, but they can change to increase or decrease the normal GFR in response to changing conditions. Complete table 5.1 to indicate whether the GFR is normal, increased, or decreased for each combination of arteriole diameter shown. The first row has been completed for you. Now let's look at this very carefully and try to understand it. Why are the answers decrease, decrease? Okay, now if you look at it, I've drawn this. This is the afferent arteriole and this is the efferent arteriole. Normally, this is the larger one and this is the smaller lumen. Now, in this situation, they have said the afferent arteriole decreases, so this becomes narrower, and the efferent arteriole stays the same. So naturally, it's going to decrease because there was a pressure difference being created because the afferent was wider, so more blood was entering it and less blood was leaving it, so pressure was created. So there's going to be a decrease in pressure. Now let's look at the other situation. Now, if you look at the other situation, what do we have? It says the afferent arteriole is normal, but the efferent arteriole has lumen has increased. So now the pressure difference which was created because of the narrower efferent arteriole now is gone because the efferent arteriole lumen has increased. So there's no back pressure going to be created. So no the GFR would definitely decrease. So that is what you have to understand is that the glomerular filtration rate would decrease. Now coming to the C part of the question, after leaving the Bowman's capsule, the glomerular filtrate passes through the PCT, proximal convoluted tubule, where selective reabsorption occurs. Describe and explain how all of the glucose in the glomerular filtrate is reabsorbed back into the blood. Now, it's a very same sort of a question which has come earlier. So very similar question earlier also. Sodium potassium pumps in the basal membrane, then active pumping of sodium out of cells, then sodium concentration decreases inside the cell, then sodium ions enter the cell from the lumen. By facilitated diffusion, sodium ions co-transport glucose into cells. This is called secondary active transport because it is because of the active transport of sodium potassium pumps. This thing is secondary to that phenomena, and facilitated diffusion of glucose into blood, and for this we are responsible for the GLUT proteins. So very similar mark scheme points earlier, but of course the only thing now is for you all to actually memorize these and remember these points. Part D, state and explain three features of a proximal convoluted tubule cell that adapted to its function: microvilli, large surface area for absorption, many mitochondria. Many mitochondria. That's the important word that you need to write. You just say mitochondria, well, I'm not going to give you any marks. Many mitochondria provide ATP for active transport. Tight junctions prevent leakage, or the substance must pass through these cells. And many transport proteins for ions, glucose, and amino acids. Now, if you look at the tight junctions, now what are the tight junctions? These tight junctions are here. This is a tight junction. If you look at it, this is a tight junction, this is a tight junction, this is a tight junction. So the tight junctions are actually holding the two cell membranes very closely together. They maintain the selective permeability, separating fluids on either side that have a different chemical composition. They seal the neighboring cells together, confine the transport proteins to their appropriate membrane. So if there's a transport protein here, now this can't go this side. So we prevent migration of channel proteins. So we prevent migration of channel proteins. So channel proteins which are here should stay here. The ones which are this side should stay this side. So they shouldn't be going migrating from one side to the other. That is what happens in the PCT. Because in the PCT you have the microvilli, then you have the basal channels. So you have the sodium potassium pump on this side. Yet you don't want these to move to the front side because here you have the co-transport channel proteins for sodium and glucose, and you don't want these to be going that side. So they prevent migration of, so seal the neighboring cells together, confine the transport proteins to their appropriate members domain by acting as a diffusion barrier, can be regulated to permit increased flow of solutes or water at the barriers, paracellular transport. So this is what I want you to know about the tight junction and how the tight junctions are going to prevent, the leakage, and then all this has to come through here. It can't pass through this, as it says here, separating the fluids on either side that have a different chemical composition. Now coming to question number 3, figure 5.1 is a photograph of a section through a kidney. On figure 5.1, use label lines and letters. So where are we going to be using label lines and letters? Please do not use a ruler for a label line. And label the region of the kidney where the following are located. So loop of Henle is always in the medulla. So the loop of Henle is always in the medulla. So you will have to be, I think this is not a very clear diagram, but I would say this would be the L, because the loop of Henle is in the medulla. Then the distal convoluted tubule is in the cortex. So that would be somewhere here. Distal convoluted tubule is in the cortex, anywhere in the cortex. And then podocyte cells, they're also here. So that would also be here, P. So L is in the medulla, D and P are in the cortex. Loop of Henle will be medulla. And this will be, both of these will be in the cortex of the kidney. So that's what they want you to see, whether they want to see whether you know this or not and where the placement is. So this is this part of the question. B part of the question: in a cold environment, people urinate more frequently. This condition is called cold diuresis. In a cold environment, homeostatic mechanisms keep heat loss to a minimum. Blood is diverted away from the skin so that more blood circulates around the main body organs and linked to the kidney. Suggest and explain why cold diuresis occurs. Possible answers to that was as all the blood is now going to the other organs, so there's less going to the skin. So the blood pressure will increase in the kidney. So there'll be more ultrafiltration. So therefore, the increased volume of urine being produced. Or you could have said ADH concentration in the blood decreases, so now all that water reabsorption in the collecting duct is not going to take place. So large volumes of urine will be passed out. So you have to really think, why would there be more urine? So think of all the things which results in less urine and just say the opposite of that. You see, when we have a blood is very concentrated, ADH is released, and the ADH causes the aquaporins, and then more water is reabsorbed in the collecting duct. So less reabsorption of water in the collecting duct. Just say the opposite of it all. Then the next C part of the question says the water potential of blood is maintained at a constant level by a mechanism called negative feedback. With reference to the maintenance of the water potential of the blood, explain what is meant by negative feedback. Again, same questions come in a previous paper. Now the obvious answers were osmoreceptors detect decrease in water potential. Osmoreceptors where in the hypothalamus, ADH released into blood by the posterior pituitary gland, increase in aquaporins in the collecting duct, so more water reabsorbed, water potential returns to the set point or the normal level. Now we need to remember these points. You all might remember it, but then if you don't sort of memorize it and forget it, after just learning it today and then taking the exam and then forgetting it the next day. Well, you need to remember these. You need to revise this chapter again and again and get all these points and memorize them. Now, reading the D part of the question, ADH affects the number of water channel proteins, aquaporins in the cell surface membranes of the cells of the DCT. State what happens to the water potential and volume of the urine if cells in the DCT have more aquaporins in their cell membrane. If the DCT has more aquaporins, that means the water which was going to become part of the urine now will enter the blood. So there'll be less water. So less water means what will happen to the water potential? The water potential will decrease. And what will happen to the volume? The volume will also decrease because less water is being reabsorbed. So it says what happens to the water potential and volume of the urine if cells in the DCT have more aquaporins in their cell surface membrane. Explain to you how the water potential will decrease is there's 10 grams of urea. Now previously the total volume of urine was say 100 ml and now it is 50 ml. Now the same urea and 50 ml, so it becomes a very concentrated solution, so the water potential will decrease. Volume will naturally decrease, that is of course clear, because less water is reabsorbed, so more water will enter and normalize the blood concentration level. But you have to understand is why will the water potential decrease? Now of course when you remove the water, it's become a very concentrated solution. You haven't removed the urea. The urea is the same amount. So the urea is the same, but now the water is less. So it's a very concentrated solution, so there are less free water molecules. So that is why the water potential will decrease. This finishes this second video, and I hope this has been helpful. And please leave any comments if you have any problems with any part of the syllabus. And thank you once again. This is the second video.